Hello. Okay, I'm going to show you how to set up your procedure for your lab tomorrow. We already went over what cello bias is, so you should know that. You should have this handout or something similar to it in front of you. If you don't, get that passed out now. But what this goes through is the measurements you need for all the buffers and the substrate and the enzyme. And then what you and your group are doing are just changing your condition for the enzyme. So you can see here, you're going to write down the name of your mushroom. If you're doing multiple mushrooms, you can put the names, comma, name, comma, etc., whatever you want. Remember, I'm going to bring cremini and portobello. So if you want a different type, bring that yourself. We are not going to use mortar and pestles to extract the cellobiase. I will show you how to do that on Friday, but essentially we're going to take a piece of the mushroom and we're going to put extraction buffer and we're just going to mash it with a pipette tip, and then we're going to get as much of the uh, fluid, I guess, as we can. We need at least like 250 microliters, which is actually a really small amount. So we are going to do that on Friday. You will be using cuvettes, and they're these cube-shaped chambers, and you're going to be labeling them with a Sharpie. So you will need 6 per experimental and control group. So you're going to run your control, so you'll need 6 cuvettes. And then you might have your first condition, which is just the stems of a mushroom. So that's another 6 cuvettes. And then your other experimental group will be the tops of the mushrooms, so which will be another 6 cuvettes. So you're going to have a lot of cuvettes. So make sure you're labeling them properly. DPTP is a disposable transfer pipette. So we're going to be using those. I'm also going to get out micropipettes, which is what they use in actual research labs, and you guys can use those too. But essentially what you're going to be doing is you'll have your cuvettes set up. They'll be labeled 1 through 6, and you're going to put the stop solution in each one. And what the stop solution does is it stops the reaction. So it stops the enzyme converting the substrate to its product. And we want it to stop so that we can, at that moment in time, determine how much product has been made. Okay? So what you're going to do is you put stop solution in each of the cuvettes, and it tells you here how much you're putting in, 500 microliters. You're going to have a few conical tubes, and they look like this, and you're going to label them with the mushroom types, okay? Make sure you always use a clean disposable pipette. We're going to be throwing a lot of those away, actually. So you're going to put 3 mils of our substrate, which is that nitrophenol, and we're going to put that into an empty tube. This is going to be your reaction tube. So, sorry, there's like some weird static happening in my life. So the reaction tube is where the actual reaction will occur. So in this tube, you're going to put your substrate and your cellobiase, however you treated it. And then over time, you'll add it to the stop solution to stop the reaction. So everything is happening in this tube here. Once you're ready, and I mean you have to be ready, like you're all ready with your timer, you know who's recording data, you're going to put the extracted cellobiase, where we mutilated those mushrooms, and you're going to put that in the substrate. Right when you do that, someone needs to start the timer because the reaction has started. Over time, you're going to be putting 500 microliters of that enzyme plus substrate into the the cuvettes. So you can see here there's times. So after 1 minute, you're going to put 500 microliters in here. After 2 minutes total, so 1 more minute, you put another 500 microliters, so on and so forth. The reason we do it at different times is because we want a rate, like an enzyme rate of reaction. To get a rate, we need 2 things. We need product produced over time. So we have our time. To get product produced, to actually figure out that concentration, I'm going to have a set of cuvettes that have known concentrations of product. And you're going to compare the intensity of yellow of each of your cuvettes with that. So then you can figure out, like in cuvette 4 at 6 minutes, how much product was produced. So you'll have your concentration over time. Yay! Then you're also going to be using a blank, so just testing the, it's like kind of like your control, but a clean disposable pipette. add 1 drop of the mushroom extract that you had over here, so just the psilobiase, to some buffer, and that's just to be a blank essentially. You are oh, that's the end. So that's just for one experimental group, all right? You will need to run it again if you're doing stems versus tops, or mushroom versus mushroom, or same mushroom, different temperatures, different pHs, whatever. So you're gonna have a lot of cuvettes. I suggest you split it up so 2 people can run a control, 2 people can run an experimental, and then you can all together run another experimental group. For our purposes, you should have that green experimental design sheet. We're not going to do 3 trials of everything. We're going to do 1 trial of everything. If we have time, we'll do 2. But we'll have to figure out if we have time. You need a control group. And if you want your control group, I have cellobiase, not from a mushroom. It's just lab cellobiase. So if you want your control to be lab-made cellobiase, you totally can, and then compare the mushroom cellobiase to that. Or if you're comparing mushroom to mushroom to mushroom, you don't have a true control because you're just comparing them all to each other. So that's kind of up to your team. So you need 1 control and you need at least 3 experimental groups. So if you're doing types, you're going to have 3 different types of mushrooms. If you're doing part, you can do the top, the gill, and the stem. If you're doing temperatures, do 3 different temperatures. pH, same thing, 3 different pHs. Understand if you go to a really high temperature right away, you're just going to denature your enzyme. So you want to keep your temperature increase or decreases pretty close to room temp. All right, so don't go to like 100 degrees Celsius, please. Alrighty. I think that's all. So what I need to see from you by tomorrow is I would like you to have your procedure filled out. There are some blanks here, so you need to put in the mushroom types that you're using, and make sure on the green sheet it's all filled out. On the back of that green sheet, make a list of materials if you need something not on here. I don't think any of you will, but just make sure you know and everyone in your group knows what they're doing for Friday. Okie dokie. Please wear your PPE tomorrow and Monday. See you tomorrow. Bye.